Starch is an example of a complex carbohydrate and natural polymer, a long-chain molecule made up of smaller units called monomers. The monomer units that make up starch are called simple sugars. Complex carbohydrates like starch are formed by joining lots of simple sugars together by condensation polymerization. This experiment shows the hydrolysis of starch using hydrochloric acid and enzyme. It also shows the tests for the presence of starch and simple sugars. First, a water bath is set up using a beaker of water and a Bunsen burner. The water is heated until it boils. Two test tubes labelled 1 and 2 are placed in the test tube rack. The labels need to be at or near the neck of the test tube, otherwise they will come off during boiling. Starch solution is added to each of the test tubes using the 5 cm cubed syringe. Then 1 cm cubed of dilute hydrochloric acid is added to test tube 1. And 1 cm cubed of water is added to test tube 2. This will be the control. The test tubes are carefully transferred into the boiling water for 10 minutes. The boiling can be controlled using the Bunsen air hole if needed. After 10 minutes, the test tubes are transferred to the racks using a test tube holder and left to cool for 1 minute. The water is left boiling as it will be used again later. A tiny amount of sodium hydrogen carbonate is added using a spatula to only test tube 1. This will neutralize any unreacted acid. This process is repeated until no more bubbles of gas are produced in the test tube. The solutions can now be tested for starch and simple sugars. Iodine solution is used to test for starch. A small amount of liquid from each test tube is transferred using a pipette to an individual dimple in the dimple tray. Then 5 drops of the brown iodine solution is added to each and the colour recorded. The brown colour observed for the sample from test tube 1 means no starch is present. The blue-black colour observed for the sample from test tube 2 shows that there is still starch in the sample. To test for simple sugars, 20 to 25 drops of the blue Benedict solution is added to each of the test tubes. The test tubes are added to the boiling water bath, which can now be turned off. When any colour change occurs, the test tube should be removed using the test tube holder. The test tubes should not be left in the water bath. a brick red coloured precipitate is observed in test tube 1. There is no change to test tube 2. The hydrochloric acid has hydrolysed the starch in test tube 1, producing simple sugars. The next experiment is the enzymatic hydrolysis of starch using amylase. In this experiment, the water bath is set up at 37 degrees. 2 test tubes labelled 3 and 4 are placed in the rack. 5 centimetres cubed of the 1% starch solution is added to each of the test tubes using a syringe. Then 1 centimetre cubed of the amylase solution is added to test tube 3. And 1 centimetre cubed of water is added to test tube 4. This will be the control. The test tubes are then placed in the water bath and held there using clamps. These are left for 10 minutes. As in the acid hydrolysis experiment, the presence of starch or sugars can be determined. Some of the sample from each test tube is transferred using a pipette to an individual dimple in the dimple tray. Then, 5 drops of brown iodine solution is added to each and the colour recorded. A brown colour indicates there is no starch present in test tube 3. A blue-black colour indicates that there is starch present in test tube 4. To test for the presence of simple sugars, 20 to 25 drops of the blue Benedict solution is added to each of the test tubes. The test tubes are added to the boiling water bath, which can now be turned off. When any colour change occurs, the test tubes are removed. An orange-coloured precipitate is observed in test tube 3. There is no change in test tube 4. Both hydrochloric acid and amylase can be used to hydrolyse starch into simple sugars. Starch is a condensation polymer made up of simple sugar monomer units. Starch molecules are too large to pass into the bloodstream and are therefore hydrolysed during digestion by acid and enzyme.